Hello, everyone, and welcome to the TMA Ask the Expert podcast series. Today's podcast is entitled Q&A on Study to Investigate the Safety of the Transplantation of Human Glial Restricted Progenitor Cells into Subjects with Transverse Myelitis. My name is Chrissy Dilger, and I will be moderating this podcast. The TMA is a nonprofit focused on support, education, and research of rare neuroimmune disorders. You can learn more about us on our website at myelitis.org. This podcast is being recorded and will be made available on the TMA website for download via iTunes. During the call, if you have any additional questions, you can send a message through the chat option available with GoToWebinar. For today's podcast, we are pleased to be joined by Dr. Benjamin Greenberg. Dr. Benjamin Greenberg received his Bachelor of Arts degree from Johns Hopkins University and his master's degree in Molecular Microbiology and Immunology from the Johns Hopkins School of Public Health in Baltimore, Maryland. He completed his residency in neurology at the Johns Hopkins Hospital and then joined the faculty within the Division of Neuroimmunology. In January of 2009, he was recruited to the faculty at the University of Texas Southwestern Medical Center, where he was named Deputy Director of the Multiple Sclerosis Program and Director of the new Transverse Myelitis and Neuromyelitis Optica program. Dr. Greenberg is recognized internationally as an expert in rare autoimmune disorders of the central nervous system. His research interests are in both the diagnosis and treatment of transverse myelitis, neuromyelitis optica, encephalitis, multiple sclerosis, and infections of the nervous system. He currently serves as the director of the Neurosciences Clinical Research Center and is a Cain Denius Foundation Scholar. Welcome and thank you for joining us today.
It's my pleasure. Thanks for having me.
So I think it would be great to just start off with just kind of explaining what the study is all about and the purpose of the study.
So this study will be the first use of stem cells directly injected into the spinal cords of individuals who have been paralyzed by transverse myelitis. And in the world of clinical research, we separate human studies into several different phases, referred to as phase 1, phase 2, phase 3, and even phase 4 studies. And this trial will be a phase 1 study. What that means is it's a small trial whose primary goal is to establish the safety of the intervention. While we all want therapies that are efficacious, we are required to show both safety and efficacy. So we split up the requirements over the course of the phase 1, 2, and 3 studies. In a phase 1, its primary goal is to establish safety.
Awesome, thank you. So the study mentions that it's using Q cells. Can you just explain what exactly Q cells are?
Absolutely. So the Q cell is the name given by the company, Q Therapeutics, that derived the cell. These are a human-derived stem cell that has been grown down a lineage of what's called a glial restricted precursor cell. And let me translate that into English. So we have known in science for decades that embryonic stem cells can turn into any different type of cell for the human body. It can make muscle, it can make liver, it can make a tooth, it can make spinal cord. And the key to stem cell research has been learning how to control the development of these cells. Because if I transplant stem cells into the brain of a patient with Parkinson's, they don't want those cells to turn into muscle or heart cells. They want them to turn into brain cells. And if I'm transferring stem cells into the spinal cord of a transverse myelitis patient who has lost the myelin around the axons of their cord, then they want the cells to become myelinating cells and not turn into muscle or heart or something else. So what went into a large amount of work on the part of Q Therapeutics was controlling the development of these stem cells such that they would become a specific cell type called a glial restricted precursor cell. This is the type of cell that can turn into an oligodendrocyte, which is responsible for making myelin. And when tested in animal models of mice that lacked myelin, these Q cells were able to go in, migrate through the nervous system, differentiate into the oligodendrocytes, and create new myelin around the axons that have been left naked in the mouse.
Okay, so that seems very interesting. Our next question is about participation. So we've gotten a lot of interest in this study, and I just wanted to go over what the inclusion criteria for participants is.
It's a great question. We get it a lot. And from the outset, I want to direct all listeners to two different sources in order to stay current with the study and with inclusion-exclusion criteria and procedures. So the first is our partner, the Transverse Myelitis Association at myelitis.org. We'll have regular updates and links to study-related documents, procedures, or instructions. And then for all clinical research in the United States, studies are listed at the website clinicaltrials.gov, which is a wonderful website where you can search by your zip code, you can search by your diagnosis, you can filter, and it will pull up all the research studies that are going on relative to whatever search criteria you put in. So today, if you went to clinicaltrials.gov and put in transverse myelitis, a lot of different studies would pop up, including this one, looking at the safety of Q cells in individuals with transverse myelitis. And at that website, on the page, are a list of what's called inclusion-exclusion criteria. And let me explain what these are in general. So whenever we design a clinical trial to test an intervention of some kind in patients, maybe it's the use of aspirin to prevent strokes in diabetics, or maybe it's the use of stem cells in people with paralysis. We are required to list the criteria of who is eligible for the trial and who would be excluded. And those criteria are decided upon based on the scientific question being asked, based on the population that's required to answer that question, based on the ethics of trial design, and based on issues related to safety and oversight. So, for example, an inclusion criteria of most interventional trials is the ability of a participant to understand the purpose, the risks, and the potential benefits of a study and provide informed consent. So we would require our participants to be able to give informed consent. We would require individuals to have a certain diagnosis. In this case, they need to be diagnosed with transverse myelitis. We are not studying the effects of these stem cells in individuals who have had traumatic spinal cord events. We're focused on individuals with transverse myelitis. So the inclusion criteria states that. It is usual and customary to set age ranges. The phase 1 studies are almost always in adults, and that is the case here. So our trial, you have to be 18 years of age all the way through 70 years of age to qualify to be in the study. And then we move into study-specific inclusion criteria that are going to be very unique to the questions that are being asked. So in the case of this study, we not only require people to have been diagnosed with transverse myelitis, we require them to have tested negative for the anti-aquaporin 4 antibody, the NMO antibody, because we are looking for individuals who've had isolated transverse myelitis and not transverse myelitis secondary to neuromyelitis optica. Individuals can't have a diagnosis of multiple sclerosis or other autoimmune disorders. And in the case of this trial, individuals need to be more than a year out from their event, but less than 10 years from their event. And I'm happy to explain why we included that. We also included a criteria that they have to have been stable for the last 3 months in terms of their neurologic function. And the goal of those two criteria were to be able to assess change over time. If we were to enroll someone who was 1 month out from the transverse myelitis and we injected these cells and 6 months later they were doing a lot better, we would never be able to know, was it because of the cells or because of the active rehabilitation they were doing and the natural repair mechanisms of the human body, because the majority of our patients thankfully recover, but not all. And so the time requirements were there to make sure that we could judge the impact of these cells, whether it be beneficial or if there were adverse events, any harm from the cells in a vacuum. And one of the most important inclusion criteria for this study was that individuals enrolling in it needed to be non-ambulatory. They could not be walking on their own or with assistive devices. And we've been asked, why is this the case? Why did we put that inclusion criteria in? And the answer is for safety evaluation. This is the first time ever human stem cells will be directly injected in the spinal cords of individuals with transverse myelitis. And in conversations with the FDA and with advisors and stakeholders, since this is a surgical procedure where we directly inject into the spinal cord, we wanted to start with a group of individuals where we felt like we would not worsen their function, or we would have a low likelihood of worsening function. So if we started with individuals who were walking around, then there is a chance from issues with the surgery or issues with the cell that we could actually lead to harm. So we restricted this study to individuals who were non-ambulatory.
Okay, great. Thank you. So how many people will get the chance to participate in the phase 1 trial?
The phase 1 trial is currently designed to enroll 9 patients.
And was there a reasoning for how many were chosen?
So the 9 comes from 2 different areas. So for a phase 1 trial of safety, you need enough patients to see if there's going to be a major safety concern, so you can't do it in just 1, 2, or 3 subjects. But at the same time, you want to be able to move from a phase 1 to larger phase 2 trials that really start to look at efficacy. And so you wouldn't want to do 20 or 50 patients in the phase 1. You want to move through it. So 9 is a reasonable number to give us a sense that the intervention is, and the surgery and the cells do not have any major obvious safety signals, but at the same time doesn't prolong our ability to move into efficacy trials. The second reason that the 9 were chosen is, in this study, through the course of the study, the amount of cells injected increase in the cohorts. So it's 3 groups of 3 cohorts of 3 patients that get increasing number of cells injected. Because one of the questions that has to be asked on the safety side is, are there safety differences between different, quote, doses of the number of cells that we are putting in?
Okay, great. Thank you. So can you explain the process of what the participants will be going through and what they can expect?
Yes, and it all starts with, for any trial, it starts with a screening phase. And the screening phase of a study is once somebody provides informed consent, there's testing that happens to make sure that individuals meet the inclusion-exclusion criteria. So, for example, a person entering this trial can't have certain health conditions like uncontrolled high blood pressure, or they can't have diabetes or liver disease. things that may put them at increased risk for having health issues pop up during the course of the trial. So during the screening phase, participants undergo testing to make sure that they don't have any of those conditions that might take them out of the trial. Once somebody passes the screening stage of the trial, we then hold a baseline visit. And this is to do MRIs and physical exams and physiology studies to really see how they're functioning neurologically, to set their baseline. And then individuals would be scheduled for surgery. Now, this study requires a surgical implantation of these cells. And what happens is my partner here at the University of Texas Southwestern, Carlos Bagley, in collaboration with a neurosurgeon at Emory named Nick Boulos, who actually developed the device that is used to implant the cells. An individual is taken to the operating room, an incision is made in their back, and the spinal cord is, the bone and tissue layers around the spinal cord are opened up so that the surgeons can visualize the target within the spinal cord, and then a series of injections directly into the area that was affected by the transverse myelitis is done in the operating room. The person would then, everything would be sewn up, and they would be brought and kept overnight for monitoring in our intensive care unit, which is standard for anybody undergoing spine surgery. And then they're kept in the hospital for anywhere from 1 to 2 or 3 days, depending on how they're doing, just like if they had gone to have surgery for another reason. From that point, they're discharged to home, and they have regular follow-up visits with my team in the neurology department over the next 9 months. Now, during this time, there are medications that the individuals will agree to take. These are immunosuppressants because putting these cells into a human is the equivalent of doing an organ transplant. And so in order to prevent rejection of these cells, individuals will take oral medications for several months to reduce the risk of there being rejection of these cells. Over those months of visits in the clinic, individuals will agree to do physical exams, MRIs, physiology studies, and blood work, all looking at the safety of the implantation procedure. Now, obviously, we are going to be looking for any signs of efficacy. We would all be thrilled to see individuals showing neurologic improvement after the implantation. But the primary goal of the study is to ensure that this procedure is safe, that we don't see any adverse events. Once we are done with the follow-up over that first year after the surgery, we are asking patients to agree for regular contacts spread out over several years after the study. Some of these would be in person, some of these would be by phone, much more spread out with less interventions. But we want to track over the long term to make sure we don't uncover any safety concerns. And also, obviously, we'd love to see efficacy over time. So this is a pretty involved study. We make individuals and their families aware that around the time of surgery, they need to commit to be near us in the Dallas area for that whole period of time. And then they have to have the ability over that 9 to 12 months after surgery to travel back to Dallas on a regular basis for the assessments and testing that we need to do.
Great, thank you. And you kind of went into this a little bit, but I just wanted to make sure we covered everything with the outcomes. So what are the specific outcomes the study will look at?
So our number one outcome is around safety, what's termed treatment-associated adverse events. And in this study, we break it down into 3 big categories. So the first is around the surgery itself. We want to make sure that there aren't surgical or post-surgical complications that occur. Bleeding, infections, anything related to the surgery itself. The procedure for opening up the spine and looking at the spinal cord is a very standard surgical procedure. Our colleagues in neurosurgery do that all the time. Injecting into the spinal cord, which has been done in other research studies, has not happened nearly to the same degree as just opening up the spine. So we're watching very carefully to make sure that the injections themselves don't lead to any bleeding or infectious issues related to the surgery. The second category of safety we're watching very closely is the cells themselves. Do we see any evidence of the cells inducing inflammation in the cord? Do we see any evidence of them forming cysts or things that should not be there? Do the cells behave the way we expect? And we'll be using MRI and physiology studies to try and understand that. And then finally, relative to safety, we're tracking the safety of the oral medications that we're using to prevent rejection. Now, all of these drugs are FDA approved. They've been used for many years in organ transplant. We feel very comfortable with the dosing and the combinations, but they have not been used in this combination or dosing in transverse myelitis patients for a stem cell study before. So we're going to be monitoring to make sure we don't see any unique toxicity in our patients. We don't expect it, but we'll be monitoring. Now, we do have a series of secondary endpoints that we're monitoring relative to efficacy. And obviously, we're going to be doing physical exams to look for changes in sensation or motor control. And we'll be doing physiologic studies to look at the speed with which signals are transduced through the spinal cord. These are electrophysiology studies that are standardly used in clinics across the country.
Okay, thank you. And so you were talking about safety and how that's the most important outcome you're looking for in the study. So the risks associated with participating, what were to happen if one of the outcomes were negative in one or more of the patients?
So whenever there's an adverse event in a study, it gets reviewed at multiple levels. So I, as a principal investigator, and my research team report the adverse events to our local institutional review board. And also we set up a separate, outside the institution, data safety monitoring board. So these are clinicians who are not directly involved with the university. They're not directly involved with Q Therapeutics. These are individuals who are there as an external check. And any adverse event is reviewed by myself, by the Data Safety Monitoring Board, by the Institutional Review Board, and decisions are made as to whether or not the adverse event was related or unrelated to the intervention. Let me give an example, because some people, this helps show the range of reporting that happens. So once somebody signs informed consent, they are officially enrolled in the study. Whether they've gotten the surgical procedure or not, they are in the study. Such that if somebody, while getting onto the MRI table for their baseline visit, falls and injures themselves, that is an adverse event as part of the study. And we would sit back and say, well, that's silly. It was a fall. So it would be designated as unrelated to the stem cells. The stem cells didn't cause the fall, but it still gets reported as an adverse event. Just like if somebody has a car accident during a study, we report car accident during study, and we have to decide whether it was related or unrelated. The core of the safety is around watching for adverse events that in any way, shape, or form could be related to the intervention. So if somebody after the surgery has evidence of an infection at the site of the surgery, that would be an adverse event related to the procedure, and we would report it as such. Independently, when there are adverse events related to procedures, the Data Safety Monitoring Board reviews them and the Institutional Review Board, and recommendations are made on whether or not to change study procedures, or if patterns ever emerge that there is a pattern of adverse events, a non-random pattern of adverse events related to a study, the Data Safety Monitoring Board, the principal investigator, or the institutional review board has the ability to pause or even stop a study if there is concerns that safety is not going to be able to be managed. And so we have multiple levels of review looking at any adverse events, giving advice on whether or not we should amend the protocol or, in the worst case scenario, pause or halt the protocol.
Okay, thank you. So if the phase 1 trial is successful, what would a phase 2 trial look like?
That is a great question, and there have been lots of conversations on phase 2 trials and how would it be different than a phase 1. I'm going to give examples of different versions of phase 2 trials, but from the outset, I want to be cautious and let people know that decisions about this have not been made, partly because we need to see what happens in the phase 1 study. But here are some examples of what a phase 2 study would look like. So a phase 2 study might be the same intervention, the same cells, but we enroll people who are within a year of their event. A phase 2 study may be the same injections, the same cells, but we enroll people who are ambulatory, people who had transverse myelitis and had an incomplete recovery, and we are looking to augment function. Maybe somebody who's using a walker to ambulate, and we want to see if we can have efficacy to get them to walking independently, or people who have ongoing bowel bladder dysfunction, and we want to see if we can fix their bowel bladder function. So the phase 2 may change in terms of both who we enroll from the timing perspective and who we enroll from a functional perspective. The 2 other changes that may occur might be relative to age. We may look at going below the age of 18 and including pediatric populations in the next phase. And then finally, the area of the cord that can be treated. I didn't mention in the inclusion criteria for this trial that we are enrolling individuals who have a lesion in the thoracic or lower portion of the cords. Individuals who have lesions in the cervical, the high neck part of the cord, we are not enrolling. And the reason for that was, again, around safety. It's in the neck that there is the neurologic control over the muscles of breathing. And there was concern that if we were seeing adverse events related to the surgery of the cells in the area of somebody's breathing apparatus, we may cause significant harm. So the first 9 patients who enter would have injections in the thoracic cord. But in a phase 2 trial, as we get good evidence that the cells and the procedure are safe, being able to expand and look at individuals with cervical cord involvement would be an option. So the phase 2 trial could have a variety of changes, and we use our experience and our data from the phase 1 to inform all of us, clinicians, scientists, the FDA, institutional review boards, patients and families, all of the stakeholders. We take the data from the phase 1 to teach us about what makes the most sense for a phase 2 trial.
Okay, thank you. And we did receive several questions asking about how long the phase 1 trial will last and if the phase 1 trial needs to end before phase 2 can begin.
So the phase 1 trial, it has 2 parts to it. The primary follow-up of patients who are enrolled goes on over 9 to 10 months. And so we have to get the last patient done with that primary follow-up to call the phase 1 trial completed. And those patients will then enter a long-term follow-up, but that data is additional. It's not necessary for designing or launching of phase 2 studies. And so the launch of a phase 2 study, while in general it does not require the phase 1 to be completed. Usually, phase 1s are completed before the phase 2 launch, but it depends on the data we're getting. If we get to the middle of the phase 1 study and things are going exceptionally well, and we have no safety concerns, and if things were amazing and we saw efficacy occurring, I can guarantee people will be sitting down and discussing how we should accelerate the program and either expand the phase 1 or add a phase 2. But in general, and that, again, this is a very just general clinical research statement, in general, we complete phase 1 studies before launching the phase
2. Okay, thank you. And we just got a question in about the surgery. So someone was asking when the first of the 9 surgeries will occur.
Yes, so we're getting close. So we are in patient what I call pre-screening mode. We have our IRB approval. We have a portal that has been launched, and there's a link to it via the Transverse Myelitis Association website, the myelitis.org website, where people can go in and answer a series of questions based on the inclusion criteria and get feedback on whether or not they qualify on high-level parameters. We were thrilled to see a robust response from our partners. A lot of people have gone online and been filling out the questionnaire, and we are now gathering medical records and imaging on those individuals so we can start to set up the screening visit.
Okay, thank you. And we also got a question asking. Oh, sorry.
I'm sorry. So we were going in order, people responding, and once we get into screening and we start knowing that people have passed screening, we will look to set up the first data surgery. My guess is it's not going to be before Labor Day, but I think somewhere in that range is when we'd be looking for first surgery, so sometime after September 1. But it all depends on how the screening process goes. I've been involved in clinical trials before where we had a long list of people who were eligible, and then we started to do blood work and surprised individuals by finding mild liver issues or mild blood count issues, things that excluded them from the trial. And so until we get people through the screening, we don't know exactly when that first surgery will happen, but it's going to be this year. The question is, how close to Labor Day will it be?
Okay, thank you. And we also received a question asking if you are selected for the phase 1 trial, is it possible to be selected for phase 2 as well?
So that's a good question. In most studies like this, most of the time the answer is no, that if you've been exposed to an experimental procedure, they tend not to enroll that population in another test of the experimental procedure. But no decision about that has been specifically made in this case. But as a generality, usually it is an exclusion for subsequent interventions under a research protocol.
Okay, thank you. So there's a lot of talk about, a cure for TM. Would you consider this study looking for a cure, or how would you classify it?
Well, I am naturally a cautious and skeptical scientist. I don't like to get too far out ahead of my skis around things like that. I would love for this to reverse all dysfunction and symptoms that were caused by transverse myelitis. I think that's going to be a stretch. I think So people ask me, what's a home run? What's success? For the phase 1 trial, success is showing safety. A grand slam is showing some evidence of efficacy, improved motor function, improved bowel-bladder function, improved sensation. That would be huge, because the moment we get a sense that the transplantation of these cells into even some individuals leads to an improvement of function, we can really roll up our sleeves and perfect patient selection and timing. Do I think there are individuals out there for whom, if we knew the exact right person and right time to inject these cells, we could prevent disability from TM? I think there's a good chance of that. That's why we're doing the study. Do I think this will reverse all symptoms in all patients? Unfortunately, no. I think there is a heterogeneity of damage that is done patient to patient in transverse myelitis. And unfortunately, our imaging techniques and our clinical tests aren't good enough yet to tell me who's who might respond, and who will be resistant to this type of therapy. So I can't call this, and would never call this, a cure. Definitely not for everyone. Would I like to help a lot of people, or even cure one person? I would be beyond elated to show that we were having a positive benefit for anyone.
Okay, thank you. We did just get a question about the cells, and can they repair damaged axons or just myelin?
Yeah, that is a great question, and it goes back to those embryonic cells. Since these are glial restricted precursor cells, that glial term means they do not turn into neurons with axons. And so we do not expect for these cells to help or cure individuals who have had a complete transection of axons. These cells will be most beneficial, based on our animal models and our experience and the theories, will be most beneficial to individuals who have intact axons but lack myelin. Now, in any given individual, our best guess says there's some of each going on, that there's an individual who has lost axons but also has intact axons that could benefit from this. So no person has 100% damage in one flavor.
Okay, thank you. And as a follow-up, is there a way to know whether a person has had complete transection of axons?
No, there is no way to know for sure.
Okay, thanks. So we have another question from our community about the 10 years out criterion. So this person says they have been told that the further away from onset of transverse myelitis, the more likely for scar tissue to form at the site of the lesion. Is there any concern that the glial cells will not be able to do their intended function if the patient is 5, 7, or 10 years from attack?
Yes, it is definitely a theoretical concern. It's worth noting that the view of scar formation over time in TM is theory-based. We have no human pathological studies, we have no autopsy studies, and we have no imaging data to tell us whether or not that's true. We suspect it, but we don't know. And so we allowed individuals up to 10 years out to be enrolled because nobody knows what the potential is for repair that far out. So in the phase 1 study, we wanted to be broad because, again, this is about safety, not efficacy. And so we wanted to make sure we were inclusive of people in a broad range while assessing, are these cells safe to implant surgically?
Gotcha. Okay, thanks. So since the study is only in transverse myelitis, how may it help people with neuromyelitis optica, MOG antibody-associated disease, acute disseminated encephalitis, or multiple sclerosis?
So this study is designed to inform us about the safety of these cells in the human nervous system and the transplantation. If we see they're safe, and if we hit that home run and we see any efficacy, then these cells absolutely have a role to play for treating individuals with neuromyelitis optica, acute disseminated encephalomyelitis, multiple sclerosis, anti-MOG antibody-mediated disease, because in all of those conditions, there will be patients who could benefit from remyelination therapy. And so we are starting in transverse myelitis, but for sure, if we see the signals we want, there will be future trials in individuals with other diagnoses.
Okay, thank you. And so if there is a phase 2 trial, will that only include patients with TM, or could that be expanded to patients with, say, MS or NMO?
Yeah, it's a good question. There's two versions that we could imagine for phase 2 trials. One where there's a single phase 2 trial enrolling people with different diagnoses. More likely than not, you would see phase 2 trials in each diagnosis somewhat separately, but that question is not yet answered. So whether or not there'll be multiple trials in a diagnosis-specific way or one big trial, we don't know yet.
Okay, thank you. We did get a specific question from someone in our community. They say, I have a spinal cord stimulator, which I no longer use. Would having it removed increase my chances of participating in the upcoming trial? They said they're going to have it removed regardless, but we're just wondering if it could increase their chance for participation.
Yeah, so having a spinal cord stimulator is not an exclusionary criteria for the trial, depending on two things. One is the trial does require individuals to be able to undergo MRI. So as long as there was not a issue with that stimulator and MRI. And the second is where the leads are placed. If they are in the area where the surgical site would be, that would be an issue that would hold somebody out of a trial. But the simulator in and of itself, just being there, doesn't take somebody out of the trial.
Okay, gotcha. So we also received a question about Q Therapeutics. So are there other companies besides Q Therapeutics that are working with the same or similar type of stem cells? And are there any other clinical trials in the works?
So there are a variety of groups that have worked on stem cells in different ways, including glial-restricted precursor cells. To my knowledge, none of those groups are launching transverse myelitis studies at this time. But there are a variety of groups with different cell types out there. But like I said, to my knowledge, this will be the first and currently only study moving forward in transverse myelitis.
Okay, thank you. So let's talk about funding. How is this study being funded?
That is a great question because we always work hard to achieve funding, and this is a true collaborative effort. So first, the majority of the funding comes from the University of Texas Southwestern, what's called Conquer program, and we had a donor from our community step up to put up the funds to cover all of the costs that would be acquired here at the university for conducting the trial, which is the bulk of the cost of a trial. All of the surgeries, all of the MRIs, the hospital stays, the medication, all of that is covered by a very generous donor at the University of Texas Southwestern, and we are extremely grateful. The cells that will be used are being supplied at no charge by Q Therapeutics. And then the Transverse Myelitis Association, in collaboration with Q Therapeutics, is putting significant funds into an account cover costs for entities outside the university. So, for example, when doing a clinical trial, there is the need to have that data safety monitoring board. There is the need to have a data coordinating center for the collection of data in a FDA compliant manner. There is the need to have regulatory oversight. And those costs are being footed largely by the Transverse Myelitis Association, with some funds from Q Therapeutics going into that account as well. And in addition, the Transverse Myelitis Association has looked for ways to help support patients and families who may have to travel to be part of the trial. And the Transverse Myelitis Association launched what was called the Eclipse Fund in honor of our matriarch, Pauline Siegel, who we were heartbroken to learn of her passing and miss her dearly. And in her honor, the Eclipse Fund was initiated so that our community could raise funds for research. And some of those funds are used to help support the aspects of the trial I just outlined. So this took and is taking a real community. There are still needs that the Transverse Myelitis Association is looking to cover. And so we are very grateful for everybody in the community who donates and helps support those efforts. And this is, in a way, a really nice example of a community. So a major donor at UT Southwestern, a large community of donors through the Transverse Myelitis Association, and a corporate partner, Q Therapeutics, coming together to do a trial in a rare disease that would never get funded just by going to the NIH or going to investors to a company to sponsor. This trial is happening because of the community.
Thank you. And if anyone has any questions about how they can help support the Eclipse Fund or the Transverse Myelitis Association, you can email us at info@myelitis.org, or you can go on our website, myelitis.org/donate. During we just got this question in, during the phase 1 trial, would you have to stop other therapies or treatments such as physical therapy or IVIG infusions if you were a participant?
So there is no exclusion for physical therapy. There is no exclusion for medications. IVIG depends on why it's being given. So if somebody has an autoimmune disorder for which they're using IVIG, it may be the autoimmune disorder that excludes them from the study. So it would depend on a person's individual history. But physical therapy is not prevented. In fact, we want all of our patients continuing with physical therapy regardless of the trial. So that would not be an exclusion.
Okay, awesome. Thank you. So somebody asked about, and you kind of touched on this when we were talking about funding, but someone asked about legislation and if there's any kind of legislative restrictions on this type of study involving stem cells.
Yes and no. So it depends on where you live. Some states have certain legislation. And then we've all kept up with the news of how the federal government is handling in different ways stem cell research. So human-derived embryonic stem cells, which have been the focus of a lot of controversy over time. In the state of Texas, there is actually legislation that limits the use of those cells in research. But there was a proviso in the legislation that said if the cells existed before a certain date, they would be allowed, basically a grandfathering in. And the cells we're using in this trial existed before that date. Now, that said, moving forward, as we talk about phase 2 and larger studies, laws such as these will play a role in limiting our ability to pursue further studies. And so the Transverse Myelitis Association and scientists around the country have worked to advocate to keep our ability to do research going, with remembering the sensitivities that go into the discussion around the cells and anything that involves human cell research.
Okay, thank you. And we did get a question in about how you are picking the participants. So is it first come, first serve? I mean, we can imagine that there'll be more than 9 people who meet the inclusion and exclusion criteria. So just what is your selection process like?
Yeah, so we struggled with this, and we abide by all of the national and international guidelines relative to subject selection, and the idea is there needs to be an even playing field, which is why all studies get listed at clinicaltrials.gov, such that anybody has equal access to the same information around the study. We've partnered with the Transverse Myelitis Association for making people aware, and launched just recently that web portal, which is the first step in the screening process. It's a pre-screening process. It's a self-administered questionnaire where an individual at home can see if they have the basic features, not all, but the basic features that would allow them to go into screening. And so we're using that portal with the date and timestamp that goes with it to basically create the queue, the line of interested individuals. And so in a way, it is the first come, first serve, but you don't have to physically be here in Dallas. You don't have to be my patient in order to get in that line. We wanted it to be a level playing field. And so we encourage people to go to myelitis.org to answer those questions. And if individuals pass that first set of questions, we will be contacting them shortly. These contacts have not started yet, so if you're on that list, you haven't missed anything. We're still going through the responses because there were a lot. You will be being contacted either way, whether you qualify or not, and if you do, what the next steps are. And we will literally go in order until we find 9 individuals who are willing and able to participate and pass the full screening. And so I expect for us to go through actually several folks. It won't just be the first 9. We're going to go much further than that to find the 9 who actually do the study. Because as I mentioned earlier, sometimes we get surprised. We do blood work and find an abnormality that takes somebody out of a study. So we won't know till we complete the entire screening whether or not somebody actually qualifies. So if you are interested, going to myelitis.org and then doing that questionnaire is the way to go.
Okay, thank you. So we did get a question about if you are participating and an adverse event does happen through your participation, would UT Southwestern and the trial take care of the healthcare for that situation, or would the person have to go back to their own doctors in their home state and have it taken care of there?
So as part of the trial will cover all costs associated with the actual study. If there was an adverse event as part of the study, that would be financially the responsibility of the individual. And it has to do with the ability of, we can't foresee everything, and so it's impossible to budget for everything. So I'll give an extreme example. Let's say somebody's taking part in a study around blood pressure control. And during the course of the blood pressure study, they have a heart attack, which may or may not be related. All of the care that's needed for their heart attack is billed to their insurance and is up to them and whichever doctors they want. whether it's the study doctors or anyone else. And so it's the same issue here. If new health issues arise during the course of the study, while we will be involved in management, those financially are outside the scope of the study.
Okay, thank you. So as we're nearing the end of our podcast, we do have one more question from the community, and then I was going to see if there's anything you wanted to add. So our last question is that you have been mentioning human stem cells. Is this the same thing as fetal-derived stem cells?
So there are different versions of human stem cells. So they encompass a lot of different ones. The only ones that have gotten clearance for trials in humans at this point are fetal-derived stem cells, which these are. The adult-derived stem cells, which require genetic reprogramming in order to make them into a stem cell, are not clear to be put into humans at this time.
Okay, thank you. So we only have a few minutes left. So I was just wondering if you had anything else you wanted to mention, or anything that potential participants should be aware of if they sign up.
As we go through with any of the individuals who express interest, who start the screening process, the first step will be informed consent, which we will go through in much more detail than I can do today, all of the different aspects of the study. And I really want to express my very sincere and humble gratitude for all of our community who step up to take part in research, whether it's a stem cell trial, whether it's agreeing to be part of a database, whether it's agreeing to be part of a study that requires a blood draw or a spinal tap. All of our community members who agree to take part in research in any way, shape, or form are giving a tremendous gift back to the community. We only get better at our treatments and evaluations by learning from our patients. And the only way to learn from our patients is via research. And so I encourage everyone, whether you take part in this stem cell study, which is very exciting, and we are just thrilled and honored to be taking the lead on. If you have the opportunity to take part in any research and are able and willing, I highly encourage you to do it. It's time, it's effort, it's a lot of sacrifice on the part of participants, but it is the key to us getting better at what we do. And we really view our patients and families as partners in the discovery process. Most of the next 100 years of discovery are going to come from translational studies where patients and families partner up with clinicians and scientists. Doing work in test tubes and cell cultures and mice is great, but our biggest discoveries are going to come from understanding human beings. And so I'm just grateful for the support we get from the Transverse Myelitis Association, and more importantly from the community that we're here to serve on a regular basis. So keep sending in the forms if you're interested in this study. We will be reviewing them. If you are not making it into the 9, don't lose faith. This is a first step of a ongoing journey that is going to lead to new therapies.
Well, thank you so much, Dr. Greenberg. I think this podcast was very informative and will answer a lot of people's questions. And thank you so much also for the work that you do and the research to come. And we're really excited about it.
Thank you so much.